Good morning. My name is Serge Przedborski, and I'm the chair of the Scientific Program Committee, and it's today my pleasure to be in presence of Dr. Rick Morimoto, who is this year the recipient of the David Marsden Lectureship. And it's a great pleasure for me because for many aspects, I think that neurobiology of disease is really becoming something that I believe is central to our progress and advance in movement disorder, but in academic medicines. And I think that, as we will discover during our short discussion today with Dr. Morimoto, really to have biologists involved in our effort, it's something critical. And so today, what I thought that we can do is to have a discussion with Dr. Morimoto. If you don't mind, I will call you Rick.
Rick, please.
And I thought that maybe what I've personally always been fascinated with, and some of my students in my lab always fascinated with, it's how great people, great scientists come to decide to go to science. I often said that at the beginning we all want to be policemen or firemen working in this or that. But science is really something very particular. And it's always intriguing to me to understand when the passion or the interest for science started. So, Rick, I was wondering if you can tell us about how that started for you.
Well, first, Serge, let me thank you and the committee for the honor of the David Marsden Lectureship. So science for me, as far as I can tell, goes back to the very beginning. Turns out I didn't want to be a rocket scientist or a policeman or a football player. Somehow my parents always brought me to the library, and I found myself interested in nature. And so as a child, I was always collecting bugs and interested in life, just, around me. Plants and flowers. And I grew up in Chicago. I was born and grew up in Chicago. And in that era, they had something called science fairs that are still popular in the schools these days everywhere. And the science fairs, I started when I was probably in the 5th grade. And I think I did something on bioluminescent. You go in the backyard on a warm, humid day, and you see the fireflies lighting up. And you sit there and say, oh, that's interesting. How do they do that? And of course, as a child, you catch a few of them. You try to take them apart. And you realize when you take apart a firefly, it doesn't work anymore. But from those simple beginnings, come curiosity that, well, how does this work? And over the years, then, as a high school student, I became so curious about science that in my senior year in high school, I decided I needed an electron microscope for a project. I wanted to examine the structure of a protist. I don't know why I got interested in this protist, but it was papers I was reading as a high school student, and I went to the University of Illinois in Chicago, knocked on the doors.
Well, you were at high school.
In high school. In high school, I knocked on the doors in my junior year of the different professors, and I would say, when they opened the door, I'd say, do you have an electron microscope? And they'd look at me, and they say, no. Finally, one man said, Dr. Howard Busey said, yes, I have an electron microscope. And I said, well, I'd like to use it. He never said to me, you're too young, this is not possible. He just, what do you want to do? And he welcomed me into his laboratory, filled with graduate students and postdocs. And here I was, I think at that time, was 15 years old, and I was fascinated. So he left me alone to do experiments. I went to the International Science Fair that year, took 3rd place in biochemistry, and met people who I've continued to be friends with in science ever since. And so I think going to college and doing science was a natural outgrowth. I went to the University of Illinois. I worked in the lab from the first day I started college. So I've always been curious about science. As it relates to how did I get to where I am now, there's really a moment. First is that I think we can all point towards specific people, mentors.
Absolutely.
And there was an amazing high school teacher who just supported me. He knew that I was not coming to high school. He knew that instead of coming to high school, I was at the University of Illinois, but he supported it. And you needed someone like that who said I wasn't being delinquent. And this one professor at the University of Illinois who just let me work in his lab, supported me. But I think the real difference happened when I was at the University of Chicago. I was very lucky. My mentor, Murray Rabinowitz, molecular cardiologist, first person to work on the mitochondrion.
And I'm sorry to interrupt, but that was what? That was when you went to college? So after high school, so I understand that you were already some kind of prodigy at the time of high school with this drive to go into science and to do electron microscopy, but then there's college.
There's college.
Right? And
so in college...
How did that happen?
So, well, I was the first in my family to go to college.
I see.
Because my parents were both put in the internment camps as Japanese Americans. So they didn't have a chance to go to college.
I see.
And so I went to the University of Illinois, and I just found a laboratory. It was actually the same laboratory that I worked in when I was in high school, because it seemed to be the natural thing to me.
Sure.
And I worked all 3 years in college. I wasn't in college very long. I was too anxious to go to graduate school. But then, again, a natural transition, working in the lab at the University of Illinois, graduating, going to the University of Chicago. And I would say, again, I got lucky. And I think all of us can look back on our careers that you can't plan it. That a lot of it's serendipity. A lot of it is good fortune. A lot of it is just being surrounded by the right people. And I found myself in an amazing laboratory doing at the forefront molecular biology when there was really very little molecular biology going on in the United States. It was one of the few labs at the University of Chicago. So that's really the beginning of sort of getting me on this whole road of excitement of science.
And when you said science, because what it's really, to me, typical of, for example, your presentation today, it's, of course, the use of very interesting models, and more specifically what you showed today, at least in invertebrate models. But it's to address a very specific question, that is, as you put it, the homeostasis of the protein folding. And that it's, of course, maybe the evolution of different steps along the line of your career development. But to a certain extent, I would say that the underlying theme to me would be neurodegeneration. On many occasions, you stressed during your presentation aging, degeneration, aside from molecular mechanism. And so that, to me, it's also something very characteristic. It's not a random theme. It's a very exciting theme, but a lot of people interested in biology will tell you that they are interested in pathfinding, in system forming, in computational neuroscience. So what led you to be attracted, one, with the invertebrate, and maybe the invertebrate is because it's the remnants of your backyard, as you said at the beginning. But the second one is, why neurodegeneration/aging?
Right. So I think the interest in neurodegeneration and then aging comes from a recognition of the importance of quality control mechanisms. That in order for any organism, if you think about us as a machine, and every other biological organism then is a machine, is that it functions as well as all the parts are efficiently utilized, efficiently replaced, which means that there has to be this really remarkable input, balance, clearance mechanism for biology. And I think it's the juxtaposition, that work that I had started as a postdoc with Matt Messelson, which is really the beginning of the heat shock response at Harvard. And really the reason why I even went to his lab, when he gave a seminar at the University of Chicago, I was sitting there in the audience. I was at that point a second-year graduate student, and I said, why should the fruit flies know what the temperature is during heat shock? What does a molecular thermometer look like? And I'm still searching for it. But what it's led me to is the realization that it's about protein damage, and then the juxtaposition that all of these very profound neurodegenerative diseases, the hallmark of pathology, that we don't understand how it causes disease, but nevertheless the hallmark as we heard in the other lectures today, is protein damage, aggregates of tau, damage caused by Parkin misfolding, synuclein misfolding, Huntington. And you then come away and realize, well, this must happen because homeostasis is not working properly. Quality control. So what can we do to restore the cell to its youthful appearance, which is how it eventually got me to the clinical thinking, and the relevance and the therapeutics is that can we take basic science discoveries, the genes we identify, and really bring it all the way to the end and say, okay, are these then therapeutic possibilities, which will not only help the neurodegenerative diseases, but if indeed there's commonality, it could actually have benefits to other degenerative diseases of aging, which I think, as a biologist, that's what I find appealing, is when you find something that's so ancient, set in biology, the imbalance leads to pathology. In the case of neurodegeneration, it's probably the most important thing we can do as scientists and clinicians is to address this, because there is nothing else. This is not like cancer or heart disease or diabetes, where there are drugs or injectables or radiation, that it's really upon us. And I think that's the other part of science, which is wonderful, is when you finally realize you have to do it. You just have an obligation to do it.
You know, one thing that I was really very struck by during your talk, because as you stressed this morning, it's really a central theme of neurodegeneration, is that with rare exception, neurodegeneration, it's a heterogeneous group that have at least in common, aside from often the occurrence of protein aggregates or evidence of misfolding, the fact that they are adult onset. And yet, if you take, for example, some of the handful example of neurodegenerative disorders that are genetic, you sleep with the enemy, meaning the bad proteins due to the mutation from day one. And basically, listening to you, basically, you can come probably to two different models. And they're not necessarily mutually exclusive. One is to think that basically the mutations skew the balance that you were talking about, but just slightly. And so you need to have a long time to have a buildup within the cells, up to the time that you will finally cross the threshold of pathology. And it happened that generally, on average, that occurs at adult age. Vice versa, you know, you have a flip side. You can also say, no, this imbalance, it's present from day one, and it's bad from day one. But our cells, as you stressed, have an arsenal of machinery that can cope and has a certain degree of buffering capacity. And aging has its toll on this buffering capacity. And this buffering capacity, basically, with time, is shrinking. Yeah. And then one day what happens is that you have this bad effect of misfolded protein that was there all the time, but that can no longer will be able to be nicely coped with. And then you come down with the disease. What do you think? And again, maybe it's not one or the other, but what's your take?
So Serge, you've hit really on a central question, but it's one that's really deeply profound because it has everything to do with evolution and lifespan. So is there a genetic plan for lifespan? Or is the genetic selection of evolution only during development? And after that, it's chaotic. Right. I think that our data is leading us more towards the latter of the two, that what keeps our cells youthful is a series of genetic pathways that keep the machines, our proteins replicating the chromosome properly, repairing DNA damage, allowing telomeres to be the proper length, preventing aberrant stress signaling pathways and oncogenesis. And that the surprise for us, as I showed in today's lecture, was that it's not a late point in aging or a midpoint, but it's actually a very precise point at the transition between a young adult and a middle-aged adult, where you now see basically a dozen different proteins losing their effectiveness, but that's coincidence with the loss of the cell's protective stress response that happens at the same time. And so obviously we have to figure out, well, is that a genetic switch, or is maybe the cost to the cell of maintaining tightly regulated genetic systems is too much, and it just simply can't maintain them. So this is the fun part for us in science, is actually to figure out what's the basis of this. But then our interest, because I think of our deep awareness of the clinical implications and the possibility that even the little things that we do can help us, guide us for therapeutics, is can we now understand what the switch is, and with what remains, restore it? And I think we can. That's the exciting part, because as a molecular biologist and a biochemist, we really look at mechanisms. And I'm hopeful that this age-dependent loss that happens, that puts our cells at risk and all these damaged proteins that are there, I think the cell actually deals with it when it's young. But at some point, the cell says, I'm sorry, I can't deal with this garbage anymore. Now what's intriguing is, of course, the same mutation in familial disease will have its penetrance at different ages. And I think that's going to be due to the other types of mutations, silent mutations, or weak mutations like polymorphisms. And I think there's growing recognition now that what makes human disease so much more of a challenge than studying it in a mouse or even in a C. elegans or a fly is we're dealing with homogeneous genetic background. And the human is anything but that. And we have to be very cautious as we extrapolate our data from model systems with limited genetic variation to humans with tremendous polymorphisms. And you can imagine those polymorphisms in some cases with some families increase the risk, and in some families it could actually be protective. Individuals that we all know of that are long-lived, that are healthy, so the combination of diet and genes. And I think at some level that's the most hopeful, because if we could understand this, then again, we're not trying to do anything fancy. We're just utilizing natural biology, recognizing that diet restriction turns out to be good, caloric restriction at some level, not the severe level, but that but the protective genes, if we could figure those out, enhance them, I'm actually quite optimistic that this can help.
In the context of diet, for example, which of course, you know, there is a tremendous enthusiasm in our society for all sorts of supplements, that there is probably two ways to see that, among others. One, it's like you discussed today, which to think that some kind of natural compound in more or less concentrate amount in natural food can modulate up or down the expression of a specific gene or a set of genes. But one thing that I was wondering that, and probably because of the time constraint you didn't touch upon, but there is more and more interest also to consider epigenetic and the role of epigenetic through the environment. How, for example, as you were saying, we have a mutation, the exact same mutation, affecting, unfortunately, in the autosomal dominant a family, and yet two brothers, for example, will be carrying the mutation, will have more or less the same phenotype, clinical phenotype, at least not to the point that's completely divergent, but still they may develop the disease at different age and with a different rate of progression, suggesting, as you said, that the genetic makeup may play a modifier effect. And to me, it could be an element of epigenetic there.
Absolutely.
And what's your take? What do you think could be the role, or how do you see epigenetic playing here as an element of the equation of neurodegeneration?
Well, this is probably among the most exciting, I think, directions. And for almost every one of these diseases, these neurodegenerative diseases in model systems, HDAC regulators, the regulation of histone deacetylases, clearly plays in. And it's fascinating because how could a remodeling of the genetic apparatus have such a profound effect unless it's resetting the clock. In some interesting way, it's resetting the clock backwards as part of a general remodeling, and in a sense, it's going back to a more youthful cell state. It turns genes on that had been turned off, maybe by purposeful age-dependent phenomena. I did touch upon it just briefly, but SIRT1, of course, is one of the most interesting of these histone deacetylases. And the fact that it directly regulates heat shock factor. It's also known that another molecular chaperone, very prominent one called HSP90 that regulates many signaling events, is also regulated by acetylation and HDAC6. So I think you're absolutely right. And this is where it gets intriguing. Could epigenetics be a way of memory of episodes of stress and caloric states? And you could well imagine that this memory actually starts at the beginning of life. And it sets actually the metabolic stress and lifespan events even in a young cell by epigenetics. And in doing so, it sets risk factors or protection to allow for this genetic variation, which we know, two siblings, twins, family members, same mutation, and the phenotypes are quite varied. So this has to happen.
Oh, absolutely.
It's so exciting. It is
Yeah.
so exciting because, again, if one's going to think about a therapeutic, it means it might not be as hard as we think it is because there's already a lot of places that you could modify without real risk.
Correct. Correct. And I think that's basically, between the line, I said that that's, to me, as you recognize, the exciting things is that, yes, we can embark in very sophisticated and targeted gene therapy, but it's true that, to some extent, if we understand better the biology of this modifier, it may be perhaps easier and more generic way to modulate the severity and therefore the disability that many of these patients may encounter because of being carrier or affected by a disease.
Yes. Well, I think I'm clearly a fan of this approach because I'm worried that if we dramatically change genetic switches, turn things on that just simply weren't on, or introduce things into cells that were never there, it might work, and in maybe very severe cases, it will give some protection or suppression. But the idea of if you understand the biology and you recognize the biology changes and you understand how it changes and you just sort of say, okay, can we restore it? So it's literally, if water goes into the ship and it goes lower, just pump the water out and it rises up. It has better buffering capacity. It can resist, it can take more people, more bad proteins, and before it sinks. Whereas if the boat is constantly taking on more water, well, guess what? You add one more person and the boat tips over.
Right. But do you think that this model of genetic targeting, I would say, or target therapies, would be the same problems that if you deal with a loss of function or a gain of function? It seems to me that in, for example, gene therapy, where clearly the loss of function of a known gene is the main cause of the manifestation, maybe there I can see that a targeted therapy could be good. For a gain of function, like for example in many of the prominent familial form of PD, whether it's LRRK2 mutations or synuclein, or in ALS for mutant SOD, it seems a little bit more complex, as you said. First of all, because we don't really know what's the nature of the gain function, but also because there could be a pervasive effect at the cellular level, and simply trying to neutralize one of the aspects may in fact rock the boat in a direction that become uncontrollable.
These are the concerns I think we have, and probably why even with the gene therapy approaches, People like Bev Davison are actually thinking about it a little bit more biologically now, where instead of trying to knock something down completely, reduce it a bit and see if you can just get it down to threshold. And I think that's very clever, because we have to think about a therapeutic maybe being applied for 30, 40 years of a person's life. And in many ways, what we're all looking for is the new aspirin, or maybe the old aspirin, a molecule that is actually very low risk, only enhances the system just enough, but as a result, doesn't cause much damage on its own. Maybe some, but not much damage on its own. You know, I'm actually very optimistic, because indirectly there's two classes of drugs out there which are given for multiple myeloma, which are inducers of molecular chaperones and stress responses. And I think we're all stunned that they actually work. So these are inhibitors of the proteasome, Valclade. It's amazing that you could block all the clearance mechanism, but seemingly myeloma cells are more sensitive to this. That's the whole point. There's different thresholds for different cell types, whether it's because it's a myeloma, because it's a cancer, it just seems to work. And of course, the other case are these HSP90 inhibitors, which also work in cancer. HSP90 is expressed in every cell, and yet you can block it, and it seems to have therapeutics. So I think there's a lot of surprises out there that the body is more resistant than we thought. The body also has its own ability to recognize imbalance. And maybe if you harness, we're getting back to this theme, that if you harness natural cytoprotective mechanisms, that the cell reads it as saying, okay, there's something wrong, so we should rebalance the system and status quo. Achieve a new status quo, versus what I call a profound, you know, I don't think we're interested in taking a cell and reversing it all the way to a very early stage. But it may be that a slight change is all you need in these damaged protein diseases.
So in listening to your analysis, where do you see that the field is going? So if you were to project yourself, let's say the life of NIH grant, so 5 years or so, what do you think along this line, the challenges that we're facing, the hurdles? And of course it can be economical, but more scientific in this case. And where do you see that we're going?
Yeah. We're on an interesting trajectory, at least in the United States. The stimulus package has, I think, wonderfully stimulated American scientists. I suspect similar things are happening in other countries throughout the world, recognizing that funding science and medicine is core to the values of every country. And anything we can do to prevent and alleviate disease, increase science and technology is outstanding. So that said, I think there's increased recognition at the NIH that protein misfolding disease is not just Alzheimer's. That's the good news. Because it's not, it means that in cancer, in metabolic disease, in kidney disease, in lung disease, the recognition that understanding Alzheimer's, Parkinson's, ALS helps inform how you're going to think about alpha-1 antitrypsin and metabolic disease. It informs on multiple myeloma. And in this case, it's probably come, almost all the really exciting data has come from people working in neurodegeneration. You know, so even for me, I got involved in this because I sat struggling at some level saying, what is the heat shock signal? Then I realized, well, what if it's protein damage? And why don't I start to study a system where everyone agrees that there is protein damage? Mutant SOD, synuclein, tau, Huntington. And so there you have then a real beginning point. You express the mutant protein and you ask what happens. heat shock, even though it's the more ancient biological stress signal, is complicated because all of biology evolved in the presence of fluctuating environment. So temperature is going to affect folding and proteins and everything else. I mean, all biochemical reactions are thermodynamic reactions, so they're all built into temperature phenomena. But at least with protein damage, then you have an opportunity to sort of say, okay, what's the signal? What's the consequence? You know the beginning moment, and you know that you haven't directly influenced membranes or DNA, but then the surprise, right? That when misfolded proteins appear, all of a sudden it affects everything else. And I think that's what I'm very optimistic to. I've seen over the past few years now many of the other institutes at NIH wanting to partner with National Institutes of Aging, National Institutes of Neurological Disease and Stroke, because they're excited that this fundamental biology is a new area to go into, and it's also, as you recognize, a little bit of a different way of thinking about therapeutics, because the history of medicine is that every disease is unique, and its phenotypes describe fully those diseases. We know well in neurodegeneration it's not so simple. The boundaries are very fuzzy, and many diseases overlap. Just the term Parkinsonism reflects a huge number of syndromes that fall under categories of which Parkinson's disease is just one of them, that in this large category. But I think that recognition is coming around to a lot of other people that, well, if you solve Parkinson's, could that inform on cystic fibrosis? Physicians would not have said that. No one would have said that 10 years ago. But if it's all protein misfolding and damage and cytoprotective responses, maybe it could. So I'm very optimistic that this is actually an important transition at the basic science level and as it relates to the disease. I'm also excited because we're having increasing number of meetings where you bring together basic scientists and clinicians. And I think this meeting, just by reflection of the talks that were given in the session this morning from Charcot and an understanding of the original description, the clinical presentation by Dr. Ling, and the basic science presentation, that you could see here we're bringing together. And it's so important because basic scientists need to know, why am I doing this? Clinical scientists need to learn the language of basic scientists so they can provide the right samples, they can provide the inspiration that's critical for basic scientists to be motivated to stay up that extra night to get it done.
Agree. I think that, as we discussed a little bit earlier, I think that really I see as a key element to our progress more manpower investment and resources investment into neurobiology of disease, where really we try to put under the same roof clinicians and scientists talking together with, of course, their different mission, their slightly different views, but really that talk together for, at the end, converge with a common goal, and that is for the benefit of our patient, the benefit of finding new treatments, and to find the basis of those diseases that remain essentially enigmatic for most of them. But, you know, thinking about this, and certainly looking at your development through your career, what do you think is the challenges that you had to go through, or even if we can touch upon if any of those happen to you, biased of our society against what you want to do? I can tell you that coming initially, I'm a molecular biologist, but coming from a clinical side, I clearly remember that I start by clinical and then be more and more attracted by biology and then go to molecular biology. And one of the biases that I had to confront almost on a daily basis was, as a clinician, I was poorly received within the biological community. It was still these images when I grew up that clinicians that are trying to do science are basically kids that playing in the sandbox, and they're not taking it seriously. And yet, I think neurobiology, it's a very serious aspect of biology. And maybe the reverse exists from your side. And of course, we all come also with our heritage, and you mentioned the painful history of your family. America, it's a very welcoming and understanding society, but still, I think that there are biased gender and ethnicity everywhere. Can you talk a little bit about that, and maybe what you can tell to our young future generation, what they should be ready for and what they should certainly not give up?
To enjoy a life of science, I think what we've both appreciated is, above all, you have to have passion. That even in the face of the occasional moments where someone says no to you or can't be done, or in a review of a paper they make a comment that just doesn't make any sense, you persist. And I think for me, I've always persisted. I've taken the path I've wanted to take. But again, I will go back to, we never do this alone. I think in all of our lives, we've been fortunate to have the right mentors, someone who's there at the right moment, who says the right thing. And that's probably the most important thing when it comes to the training of new young scientists, is that they have to have the opportunity, they have to have the trust in themselves to find the right person, the person who's going to really challenge them to be the very best that they could ever be. Not just comfortable, just someone who just understands them and is going to encourage, support, but above all, push them into the level of excellence that they deserve. There is no complacency when you want to discover, ever. I've never been bothered by, oh, well, I might not get this grant, or this paper might not be published. There's the next time, and the next time after that. And you never stop trying, you never stop looking at opportunities. You know, scientifically, in many ways, I was lucky in that I made the transition to neurodegenerative diseases after I had pretty much established myself as a molecular biologist with the cloning of molecular chaperone genes and heat shock factors and really understanding the mechanism. But I did get the most wonderful reviews in some of our very early papers. Wonderful reviews saying, how does Morimoto think he's going to solve neurodegenerative disease with a worm? Okay, I understand the comment. It's a bias. But it's also an ignorance because the person who said it doesn't appreciate biology. I'm not going to generate a clinical protocol from a worm. That is true. But I think we can inform. And I think that's been, for me, one of the most exciting aspects of the last 5-10 years, is that we're not the only lab. This is now clearly recognized as a way, in partnership with other model systems and clinicians, to solve really some of the most challenging biology in medicine. The advantage of an organism where you can study its lifespan within a period of hours, where you can study its entire aging process in a matter of weeks, allows us to do experiments that you then work with someone who's doing transgenic mice and says, I can help you design the better experiment, because you don't want to spend 5 years making 15 different crosses and complicated transgenic lines and then discover you made the wrong ones. Biology is conserved. And I think that, again, is something that's exciting. When it's not going to be a human, well, I don't think all of it's going to be a human-specific. I had said earlier that we have to be cautious not to think that what we learn in a model system tells us exactly what happens in a human. And I think there's a tendency in the clinical therapeutic world, and as it relates to pharma, to make that mistake over and over again. And I think that's where the link back to the clinicians, they're the only ones who see the phenotypic diversity, and they respect it, and they see it all the time. And I think that information needs to penetrate more and more, and we have to understand what some of that variation is. I think that offers great hope. What's the basis of that variation? Maybe that's the easiest thing to go and solve, because if two siblings get the disease but off by 10 years, so one gets it at 55, the other got it at 45, and the course is not identical, gee, you have a protective effect. That's exciting. You've just added 10 years. This person has 10 years more than the brother or sister. I think this is great.
So is it fair to say that it seems that I hear a little bit what I believe as well, which is that in the face of difficulties and bias and prejudice, persistence and passion, it's probably what should drive us. And certainly, if you do not want to face those type of difficulty, I think that science or academic medicine, it's maybe not for you, because it is a difficult, merciless path. When it's not people around you, it's simply the discipline that required a lot of rigor, that often fail and prove you, even for the most intelligent and brilliant, how stupid we are and how wrong often we are. But still, that's what we like, that's what we love, that's what we have to put all our energy, and not let anything or anyone detract from the path that we want to pursue.
Well, I've always felt that it is the greatest honor to be a discoverer, to have the honor to understand biology, what makes us live, is the greatest honor there is, and that one has to approach it with humility and yet confidence on the other hand. You have to have confidence in what you're doing, yet in the face of it to always be humble, because we don't know, but we are constantly working at it. And that's why these meetings are so important, scientific interactions, because if you stay alone, you are a genius in isolation. You have to be out there talking to people at meetings, at poster sessions, and constantly testing your ideas over and over to find out, because that's how you refine your ideas. And what once seems to be a crazy idea then becomes an important part of a field.
Rick, I can tell you that on behalf of the society, and I'm sure that on behalf of everybody who was in the room today, you have been extremely inspirational. I would love to be able, in the era of progress of technology, to be able to clone you and to have more people like you in science, in neurobiology, in clinical sciences. But I can really express my gratitude for everybody here. And again, congratulate you to be the lectureship for the Marsden lecture this year. Well deserved. I think that if David, who was a remarkable individual, would have been with us, he would have been very proud, as we all are, to count you among the recipient and to have you today here in Paris. Thank you very much.
Well, thank you very much. Indeed, it's been an honor, and I very much enjoyed sharing our thoughts and philosophies about science and discovery. Thank you.
Thank you, Rick.